All right, so we are about halfway through our hierarchical organization, where we said atoms to molecules. We took a look at specific macromolecules, and now we're going to see those macromolecules play a role in the formation of a cell. And then we have tissues, and then after that, the rest of 168 and 169 is just organ system after organ system. So this is cells, part one of, I think right now I have 5 parts, but I think I'm probably going to split that up into more. We'll see how that goes. All right, so for the intro part of cells, we're just going to talk about the cell theory. We're not going to get too detailed with the cell theory. There are 2 principal tenets of the cell theory that I want to kind of touch upon. And the first one is that the cell is the functional unit of life, which just says basically if something is alive, it has to at least be made up of one cell. Okay. I'm going to get back to this in just a second. So just remember, this is more for me to just kind of remind you of something more so than something that you guys have to write down in your notes. So I will get back to that. And then, so if all living things are made up of cells, and I made mention of this when I did the characteristics of life. So continuity of life has to have a cellular basis. So we have to talk about how the cell will reproduce, and then how the organism reproduces if that's a multicellular organism. And since this is human anatomy, it's exactly what we're going to be doing in later units. All right, so these biochemical activities of cells are dictated by subcellular structure. So this is a generalized cell. So what we're going to be doing is we're going to be doing a cheesy tour of the cell, saying, hey, look to your left, here's the mitochondria. Look to your right, here's the Golgi apparatus, so on and so forth. Not every single cell is going to look like that. This is just simply for convenience that we're going to be taking a look at all of these subcellular structures. And after we get done with this intro and then talk about how stuff passes through this membrane, then really we're just going to hit organelle after organelle talking about what it does. All right, so not every single cell will have all of these. Quick example, centrioles. Centrioles are involved in replication when a cell divides. So we're not going to see centrioles in neurons because they can't divide. Mitochondria. We're going to see a lot more mitochondria in a cardiac muscle cell than we would a skin cell. So the amount and if an organelle or subcellular structure is present, it's really predicated on what that cell does. Again, principle of complementarity, and we see that on a cellular level. And just to kind of reinforce this, when we take a look, all of these cells can come in so many different sizes and shapes. There is some sort of limit on the size of a cell, and that's because of surface area to volume. But nonetheless, then we have all of these different cell shapes. And that's, again, to just kind of reinforce what does this cell do. So not every single cell is going to have all of these structures here. Okay? So that's pretty much it for the cell theory. Now we're just going to kind of go over some cell basics here. Then we have the cell membrane, and that surrounds the cell. All of this is the cell membrane. And it's made up of proteins and lipids, and we'll get into the exact makeup of the plasma membrane in a little bit, in a couple of, maybe part 2 or 3. And composition and function can vary from one region of the cell to another, and from one cell to another, meaning what type of lipids, how many proteins, all of these things can vary. The cytoplasm is going to consist of the organelles, all of these organelles, this cytosol in here, this cytoskeleton in here. So we have the organelles, cytoskeleton, which is just kind of like a road network inside the cell. And then what's left, that fluid, is called the cytosol. Some biologists get really picky at differentiating between cytoplasm and cytosol. I'm really not that concerned about it. Later on, we are going to be talking about an ICF, an intracellular fluid. And again, it's not that critical if you understand the difference between cytoplasm and cytosol. And then outside of the cell, there's going to be some fluid, and that's going to be the ECF. Okay, so I just want you to understand what ICF and ECF is. 169, we're going to be identifying the composition, anions and cations, the major ones that are in each. So the question becomes, what is the benefit behind having all of these subcellular structures? And the benefit is just that, compartmentalization. You don't have to worry about all of these organelles right now. Just the whole premise of, if I need energy, I know that I can go to the mitochondria and get energy. If I need to package something, I know that I can take it to the Golgi, so on and so forth. So by having these subcellular structures have very specific functions, that compartmentalization has allowed for an increase in complexity. And we can kind of see that here. This is just to reinforce that. I'm not going to test you on any of these numbers or anything at all on this. This is really just to demonstrate how intracellular fluid is about half of the cell, and the rest is really just inside all of these organelles. Okay, that compartmentalization, again, and this is just for a liver cell, but that compartmentalization has allowed for an increase in complexity. And again, we're just talking about comparing our cells to a prokaryotic cell, some sort of bacteria. Okay, pretty nice and easy intro to the cell, and that's it for part 1.